Okay, so I've added a little subtitle on the top, which is Move Over Mice, and that's move over, not get out of the way, because I think that there's a role for animal models, but I also feel that there's an increasing role to have more complex models of the human condition. And so my lab's a tissue engineering lab, and I worked in regenerative medicine for a long time, and then started turning more toward these kinds of models, mainly for liver and the GI tract and others. Only a small part of my lab, but the most important part of my lab works on reproductive health. Everything in my lab, though, everyone is involved in endometriosis because, as you heard from Hugh Taylor, it is a systemic disease, and so we think about that a lot. And so what I'd like to do, I started working on endometriosis about 10 years ago, pulled in by my wonderful clinical collaborator, Keith Isaacson, who said there's really no great therapies for these patients. My niece in Atlanta had just recently gotten diagnosed. And fortuitously, right after we started working together, I got diagnosed with breast cancer. And I was immediately classified as triple negative, which is not a super good thing, but it was just 1 year of horrible stuff, and then it was done. And endometriosis is a lifelong horrible thing for many women. And what it made me realize is that we don't have great ways to molecularly classify endometriosis patients. So endometriosis is not one disease. We've heard that today, and we hear that everywhere. And so a lot of what we're trying to do in our overall program and community is think about ways that we could classify patients. And I'm going to, I put in a slide on adenomyosis. I haven't heard it mentioned much today, and you'll see in the upper right-hand corner of this slide a calibration for how little adenomyosis is studied. I've compared PubMed citations of adenomyosis. So adenomyosis is when you have endometrial glands and stroma misplaced in the myometrium, arguably hugely underdiagnosed. Katerina Exakoustas published some beautiful work showing how you could infer it from imaging studies, and now it's more commonly suspected, but we really only the tip of the iceberg on this. But you can see that a disease everyone would presume is cured, because you watch football, you see ads for drugs for erectile dysfunction has almost 24,000 papers, and adenomyosis only 2,300. So we have a long way to go, and I hope in future years we have this at the disease. So we started out with a hypothesis, and I know there's a woman here who says she's completely asymptomatic and infertility was the first way she presented. And this underscores how patients can present very differently, not just endometriosis, but almost all chronic inflammatory diseases, arthritis, et cetera. And so are there molecular mechanisms that differentiate groups of patients from each other? One way we might learn about this is from the kind of studies Stacy Mismer does in GWAS. That's from the genes up. A way we also approach it is from the top down, thinking about networks of inflammation, invasion, et cetera, that may be diagnostic of different subsets of endometriosis. And I put hypotheses. We haven't proven this, but it is something I think worth considering. It requires large patient populations. Okay, so first of all, we want to, how do we even find mechanisms in groups? And I'm not going to focus on that today. I'll just mention what we've done. But these studies on patient populations led us to think, if we find mechanisms and we want to develop drugs for subsets of patients, personalized medicines, how do we tell that they're effective? And every drug company I have talked to about new classes of drugs for endometriosis outside of hormones, you got to have an efficacy model. That's what they say over and over and over. And it has to be something other than animal models, because we have cured so many animals of endometriosis with new classes of drugs. So that's what I really want to talk about today, the tissue engineering models. Now, I will mention that the motivation for what I'm going to talk about is a whole set of studies we've done in patients with collaborators around the world, and papers still pending to be published, involving trying to look for molecular classifications. And I could be happy to discuss those with you later. So what about tissue engineering? Well, tissue engineering almost always implies some kind of 3D culture. Why would you want to do 3D culture when you can put cells in a petri dish? Well, there's reams of examples in the literature now showing how when you culture cells on a stiff substrate, like a tissue culture dish, versus an environment that's more gel-like their normal environment, that this stiffness and mechanical properties can drive inflammatory phenotypes. So this just happens to be from liver, Becky Wells' lab at Penn, showing activation of hepatic stellate cells when cultured on stiff plastic compared to a gel of the same modulus that you'd find in liver. So you can see it's getting activated with smooth muscle actin. I work a lot on liver, so I happen to have this slide. I don't have one yet for endometrium. But this is really, really important and why we want to do things in gels like matrix. Now, there's been a lot of efforts in tissue engineering of the endometrium. I'm showing two here, one from Rob Taylor's lab where they grew stromal cells and did a hormonal cycle and showed that they could, not super reproducibly, but could get breakdown of matrix and induction of MMPs. And one from work done by Bruce Leslie a number of years ago looking at co-cultures of endometrial epithelial and stromal cells. And here they used Matrigel and trying to look at crosstalk effects on proliferation and et cetera. And so these are the tools available to most biologists to do 3D culture, natural gels made from collagen and Matrigel. And here, these were both cell lines in these studies. And so to move more toward the kinds of systematic studies that can be reproduced across many labs, but also let us pull more information out, we want to move toward a synthetic extracellular matrix that allows us to manipulate the extracellular environment in a completely modular way and allows us to dissolve it at will whenever we want to release the molecules that are crosstalk between these cells and also to release the cells. And so these would be used to test complex disease mechanisms because you can look at paracrine signaling. You could ask questions about patient stratification if you get tissue banks, and you could test interventions. So 3D culture, multiple cell types in a completely synthetic defined microenvironment that you manipulate and that you can share with your collaborators and other people, anyone who wants it, to do similar studies in their labs. And so I'm going to just, and then of course for the endometrium, there's two broad classes. You could build the models of the eutopic or models of the ectopic endometrium, and I'll quickly review what we've already done in that regard using both primary cells and cell lines, where we did static 3D culture models, meaning we just had them in dishes, we didn't actually put them in a bioreactor. So let me tell you about that real quick. But first, let me say something about the sources of cells, even in just, if you're just going to study general endometrium. You can use cell lines, but in our hands, they are not really representative, surprise, of the normal endometrium. And so we get all the tissue from endometrial biopsies from our clinical collaborators. And you can get these from all kinds of different patients. None of the work that I'll talk about today is actual lesions. So a really important reason to use biopsy, primary materials is shown here. These are cytokine production data in a co-culture of stromal and epithelial cells. And what I'm showing is 3 different patients compared to cell lines, Ishikawa. For all these different cytokines, they're in groups where primary cells didn't make as much as the tumor cells, no difference, higher or only detectable. So the key thing is the blue dots from the cell lines are way below those of the patient samples. And the patient samples show quite a bit of variability. So there's going to be donor-to-donor variability, and that's something we think about a lot and we're trying to control for. So what is our synthetic extracellular matrix? I'd be happy to send anyone the publications, and we actually share the material if you're really serious about using it. It is based on a synthetic polymer, polyethylene oxide. It's activated with reactive groups, so that lets us put biological molecules that interact with cells or matrix there. And it also lets us cross-link it by putting a peptide that has a disulfide on either side, so we can cross-link it. And when you make it, it looks like a soft contact lens. Completely clear. You can do this with a pH change at cross-links in minutes. You can also do it with a UV light irradiation depending on your activation step. And importantly, we include within this sequences that are degradable by MMPs so that cells can remodel their environment. And we also have a special sequence that can be degraded by a bacterial transpeptidase called sortase. There's very few known mammalian substrates of this, so we can dissolve our matrix and get all the proteins out and do all kind of multiplex proteomics on them. So just to review what we have done in terms of the initial steps in this kind of tissue engineering is build a matrix that, a tissue that looks like a a eutopic monolayer tissue, and this is just in a trans well, so you can access the top and bottom and look at things going on at the top and bottom separately. And here, the synthetic matrix fosters accumulation of basement membrane produced by the cells. So we're looking here at this focal plane at basement membrane, and it makes a beautiful basement membrane by the epithelia. And then if we go down and look in the stroma with confocal microscopy, what we see is, again, accumulation of matrix. This is fibronectin stained green, and we can also get type 1 collagen and organized. We can add immune cells, and they will migrate in and migrate around through this matrix and ultimately differentiate into tissue-specific innate immune cells. Okay, so, and that's all been published. And so we can also start to build lesion models, and here I'm showing, this happens to be Ishikawa cells just encapsulated in this completely synthetic gel, again, having completely properly polarized with basement membrane. And here we compare co-cultures of stromal and epithelial cells, staying here, the epithelium here, the stroma, in either matrigel, which is the canonical natural matrix, very poorly defined, lots of growth factors, et cetera, et cetera. And here's our PEG hydrogel, or synthetic matrix, and we can get beautiful behavior of the cells in this matrix. And so now we can, what held us back initially was that it was hard to expand the epithelial compartment. But using the same approaches that Hans Cleaver first described for intestinal epithelial cells, we have built on publications from 2 different groups to now adopt the expansion of primary human epithelial cells in this organoid environment. And our lab has a huge sort of core facility for organoids for gut epithelia, and we're now using it for the endometrial epithelia, as I'll show in a moment. So this makes it completely possible to build a tissue bank that has not just stroma, but also the epithelia. This is a little flow diagram from Juan Necco, a fantastic postdoc who came to my lab from Kevin Osteen's lab with Victor Hernandez, my postdoc who works on the gut epithelium. The endometrial glands, you take them, you grow them in matrigel with a special medium, and they form organoids, and then we can put them in the synthetic PEG hydrogel, so it's a completely synthetic matrix, properly polarize, expand, and co-culture them with the other cell types from the endometrium. Now, I want to turn to where we're going with this. So we've gotten these tools, but it's very unsatisfying to just look at these in a little gel floating around in a Petri dish. What I'm really interested in right now, and it came up so many times at this meeting, is thinking about the early stage lesions. So we've seen movies over and over again today, or other evidence that these lesions bleed at least some of the time. And this is a paper from Ruth Lottie, who's an MIT grad, so it was fun to show this. Her daughter is now a sophomore at MIT, and I know her. So these lesions are bleeding. And so a question that comes up to me is, you know, you've got great surgeons. Let's imagine the patient can have her visible lesions removed. But microscopic lesions, I think that there are probably going to be some left behind, no matter how great of a surgeon you are, and we don't have the tools to see every single lesion. So is recurrence coming from these microscopic lesions that are left there? So for the patient today, she has excision. What happens? Why does she get recurrence? So a question is, if we could develop some kind of therapies to treat those processes that allow these lesions to come back and be so horrible, would that be beneficial? So from a tissue engineering standpoint, maybe reproducing phenomena in these early lesions would help us understand lesion progression and also test their potential therapeutics. So that's on the table. I'm just now going to show you some very speculative things and some preliminary data. This is actually out of a grant we just submitted in response to an RFA, so I'm doing something you're not supposed to do in science. Young people don't do this with your ideas, but I'm an old person, so I'm just really committed to trying to get this to work, and I'll find a way to do it. So here's what we want to do. So you've got a trapped epithelial cells here. You've got some stroma, and they're communicating with the vasculature, probably increasing the permeability, maybe even to the point it bleeds. Okay, so that will likely then recruit circulating cells, and they may just bleed out into that lesion, but they certainly could be recruited in some fashion. And this may be immune cells or stem cells, anything that's going through the circulation, SDF1, whatever's coming from these tissues might be recruiting. Okay, so that's an early step. And then you progress under cyclic hormones. Okay, so you got to have cyclic hormones. You progress to a point where these cells that get recruited recruited start to turn into things. Maybe they turn into macrophages. Maybe there's some NK cells doing some crazy stuff that NK cells do. Whatever, and then they alter perhaps the vasculature, ultimately progress to fibrosis. So the question is, can we build a little model like this in culture? And I think the answer is yes, and let me tell you how. So first of all, we know that vascular interactions with the stromal and epithelia are likely hugely crucial. This is known, and Juan Necco, when he was in Kevin's lab, built a really nice little microfluidic model where he put flow chambers together with a membrane here and had endothelium and stroma. And he showed in this model system that flow across the endothelium influenced the decidulization capacity of the stroma. So what you see here is without flow, this is the decidulization response of the stroma, and with flow, this is the decidulization response of the stroma. Okay, so this was a very wonderful model, especially for someone in a lab that's not a microfluidic lab. He was heroic to do this, and then he came to my lab where he really wanted to use higher power tools that might be available in our microenvironment at MIT. And I'm going to pass one around. I'm going to pass around this little microfluidic device because I'm going to explain it. It's actually a much smaller version than we use, but this is actually a commercial device that you can buy from a company in Singapore. It's got 3 little microfluidic. So this is a region that you can put a gel, and if you put endothelial cells and fibroblasts and certain kinds of gels, they will spontaneously organize into vessels, microvessels, that can be perfused. And along either side, you can flow medium. And in this particular device I'm passing around, the way that you flow the medium is you attach reservoirs that have different fluid heights so gravity drives your flow. So it's a slow interstitial flow. Okay, and there's a lot of other organs, so-called organs on chips that are out in the community, and you've probably heard about this. Okay, now, with the exception of the one I'm passing around, which is made out of a thermoplastic, most of them are made out of silicon rubber. So the ones out of Harvard, for example, silicon rubber, Berkeley, silicon rubber. And silicon rubber is a pretty bad material to use if you want to study endometriosis because silicon rubber absorbs everything lipophilic, including estradiol and blah, blah, blah. So we, over the past 15 years in my lab, we've been developing organ-on-chip technologies that don't use silicon rubber and have some pretty kick-ass pumps. And this is work of my colleague Dave Trumper in mechanical engineering. And we got a big program from DARPA, about $25 million over 5 years, to build interacting organs on chips. And this is just a humongous paper because we had to have 10 organs. And they're actually interacting for a month, so fluid constantly being pumped in between them and so on. And the purpose of this was simply for me, I did this because I wanted to build technology that would let me study systemic endometriosis. My DARPA program manager let me spend $1 bazillion on the tissue engineering of the endometrium. So this got done. We published the paper, 30-something co-authors. And the nice thing is we now have technology that lets us pump stuff around in a really fabulous way. But none of these organs that we used here were vascularized. Okay, so we did not do that in this program. However, my colleague Roger Kam, who's in mechanical engineering, has done this. And let me show you, and I just have pictures for the rest of this, so it'll go real quick. So his device here, so this is the device you're seeing is a smaller version of one shown here. As again, you put the endothelial cells and fibroblasts in, they self-assemble to form vessels, and the vessels are incredibly stable. So day 16, day 23, and you can perfuse them because fluid can go from here to here. You can flow cells through these little vessels, immune cells, tumor cells, yada, yada. And what that lets you do, and what his lab has been doing a lot lately, is look at trafficking of cells from the microvascular into the tissue space. So here, for example, cancer cells extravasating into a tissue space helped by neutrophils. Here's the movie of that happening, the neutrophils in red, and the cancer cells here. And what you'll see if you read the paper is that quantitatively the neutrophils help the cancer cells escape. Okay, now once the cancer cells get out of the microvasculature, they sit here and then they can grow into a tumor. Okay, and you can image all of this in real time because this isn't this little device that you can put in a confocal microscope. All right, so of course, my mind turns to trying to build lesions, and more recently his lab, and this isn't published yet, it's the work of one of his postdocs, is the tumor, which is here. If you put a tracer fluorescent tracer of this should stay inside the vessels. It stays inside the vessels till it gets over here where it's leaked out and you can't see it. So the vascular permeability increases dramatically in the vicinity of the tumor, and you can measure that and quantify it, and the stromal cells are recruited to the tumor. So these are all things that can be quantified, and these are tumors. So now we are just starting to design and build devices for lesions, and this is simply to say we have to do a lot of math to calculate what kind of flow rates and blah, blah, blah. But Juan Neco, this amazing postdoc that came from Kevin's lab together with Roger's postdoc, Gio Fedu, has recently just started to build models of the lesions combining epithelial organoids with stroma and either leaving them as is or pre-treating them and putting them into these devices. And you can see here the ones pre-treated with IL-1 beta are hugely invasive and they disorganize. Sorry, this is very recent preliminary data, like from a week and a half ago. And so these are not perfused vessels yet. We're simply just getting them in the device, and we didn't have it set up to do the flow. So this is where we are going. Okay, so the thing is, can we build a vascularized 3D lesion? I think we can. I think at this point it's a technology push. What should we do with it? And that's why I came to this meeting. I want to ask you what to do with it, and we can talk about this later. And I want to connect it to other organ systems, et cetera. I'll end there, but I want to thank my most amazing collaborator, my husband, Doug Laufenberger. This is his student, Abby Hill. He has given his time and money and everything to work on endometriosis when I've never given him any funding. And he's been a tremendous collaborator, a lot of other collaborators, and I'll especially call out the mother of everybody, Stacy Mismer, who has helped my lab immensely understand the multifaceted natures of this disease, and through her work with the WRF Effect Project has really helped the entire community. And there's, of course, many of you here to who I owe tremendous debt, and I'm not showing your pictures, but Michael Bestie, who did the epidemiology, and then other folks at MIT.